Now students, let us understand the second type of cell division, that is mitosis. Mitosis is a type of cell division in which a cell divides to form two daughter cells, two new daughter cells. Now let us look on some general characteristics of the mitosis division. First is the cell divides to form two similar daughter cells. The daughter cells which are formed after the mitotic division, those daughter cells are similar to each other. They are same. Second, in the process of mitosis or in this mitotic division, equal distribution of genetic material is done. Equal distribution of genetic material. Now here the daughter cells which are formed, in those two daughter cells, the genetic material, that is DNA, is equally distributed among those two daughter cells. Now this mitotic division can be seen in the somatic cells. It can be seen in somatic cells. Somatic cells are the cells of our body, all the cells of our body except the reproductive cells. So the mitotic division can be observed in the somatic cells of our body. Also, this mitotic division is called as equational division. Now, why it is called as equational division? It is because when the daughter cells are formed or when the daughter cells are born, those two daughter cells have the same number of chromosome as their parents. For example, suppose this is a parent cell, this is the nucleus. After the division, when the daughter cells are formed, these two daughter cells will have the same number of chromosomes. Suppose this parent cell has 23 number of chromosomes, so the daughter cells will also have 23 number of chromosomes. This cell will have 23 number of chromosomes and this cell will also have 23 number of chromosomes. So the chromosomes or the number of chromosomes is equal to the number of chromosomes in the parent cell. That's why this type of division or this mitotic division is called as equational distribution or equational division. Now this process of mitosis can also be occurred, or it happens in the two processes, or the division of mitosis or mitotic division occurs in two types of processes. Generally, cell division occurs in two types of processes, that is karyokinesis and cytokinesis. Here also these two processes are seen, that is karyokinesis. In karyokinesis, as we said, it is a nuclear division. So the nucleus which is present inside the cell, it tends to divide into two daughter nuclei. So the nucleus which is present in the cell, it divides into two daughter nuclei, and after this process of karyokinesis, the process of cytokinesis occurs. The process of cytokinesis occurs. Cyto means it refers to the cell, cytoplasm, and kinesis means division. So the division of cytoplasm takes place over here. After the karyokinesis, the cytoplasm shows a constriction in between, and as this constriction gets deeper and deeper, the cytoplasm divides the cell into two new daughter cells, or that parent cell is divided into two new daughter cells. So at the end of the process of mitosis, we get two new daughter cells, and these daughter cells, they have equal number of chromosomes, they have equal genetic material, same genetic material, that is DNA is same in both the cells, and they have two individual nucleus in them. Two separate nucleus are there in those two daughter cells, and this type of division occurs in the somatic cells. Now let us understand some terms which will be helpful for us in this chapter further. Now as you can see this diagrams over here, this is the strand of DNA. It is about 2 meters in length. So the DNA, that is 2 meters in length. Now, if the DNA is this much longer, how this DNA is able to fit in this small nucleus? It is because the DNA, it gets coiled up and it forms this structure. It forms a structure like this, and this structure is called as chromosome. So the DNA gets coiled together to form a structure that is called as chromosome. Now, how does this DNA gets coiled together? There is a protein present inside the cell that is called as histone. This protein helps the DNA to get coiled and it forms the structure. Actually, this histone protein, it holds the DNA, or it coils the DNA around it, and together it forms the small structure that is called as chromosome. Now, it is made up of two structures like this. The chromosome or the DNA gets coiled into two structures like this, and these two structures together they form the chromosome. These two structures separately, they are called as sister chromatids. They are called as sister chromatids. So the DNA gets coiled together with the help of histone protein to form chromosome, and these two strands of chromosome, they are called as sister chromatids. Now, if we talk about the reproductive cells, in the reproductive cells, the chromosomes are always in pairs. These chromosomes, they are found in the pair. This pair is because of one chromosome comes from the father and another chromosome comes from the mother. So the reproductive cells, they have two pairs, or they have a pair of chromosome, and this pair of chromosome is called as the homologous chromosomes. Now, why these are called as homologous chromosomes? Because they come from, or they are arised from the same part of the DNA. That is why they are called as homologous chromosomes. Now, we will look on to some other terms, that is haploid cells and diploid cells. Now, what are haploid and diploid cells? Now, if a cell has a type of chromosome in a single copy, that type of cell is called as haploid cell. And if a cell, it has a type of chromosome in two copies or in pair, that type of cell is called as a diploid cell. So earlier I said that the process of mitosis is divided into two processes or two stages, that is karyokinesis and cytokinesis. In karyokinesis, the division of nucleus takes place, and in cytokinesis, the division of cytoplasm takes place. Now, this process of karyokinesis is further divided into 4 different phases. These are the sequential phases, starting from prophase, metaphase. Metaphase is followed by anaphase, and the last phase of the karyokinesis process is the telophase. So these are the 4 phases in which the karyokinesis process is divided. Prophase is the first phase, metaphase, anaphase, and then the last one is telophase. Now, this prophase, it is further divided into 2 types or 2 phases, that is early prophase and late prophase. Now, first we will look about we will discuss about the early prophase. Now, let us begin with prophase. As I said, prophase is further divided into 2 phases, that is early prophase and late prophase. As you can see, the diagrams I have drawn over here. In early prophase, the nuclear membrane is visible, nucleolus is visible, the duplicated centrioles are visible. These centrioles, they get duplicated in the interphase. They are visible over here and the chromosomes are visible. Now what happens in the early prophase is that the chromosomes, they start condensing or they get condensed, and this nuclear membrane which is there, it disappears. It slowly starts to disappear. Also the nucleolus starts to disappear, and these centrioles, duplicated centrioles, they start migrating away from each other. And in the late prophase, as you can see here, the chromosomes have already condensed. These are the condensed chromosomes. They condense together and they form condensed chromosomes. The nuclear membrane is integrated and it starts disappearing. It starts disappearing and it is integrated and finally it disappears. And the nucleolus also disappears. And these centromeres, these centrosomes or centrioles, these centrioles, they start moving away from each other. They start migrating away from each other and they start moving to the opposite poles. They move to the opposite poles to each other. If one centromere, if one centriole is here, then the other will be opposite to it. In the late phase, the nuclear membrane disappears, it integrates and it disappears. The chromosomes are condensed and the centrioles, they are on the opposite sides or opposite poles of the cell. So this is the prophase. Prophase is the longest phase in the cell division or in mitosis. It is divided into two phases. Early phase shows the nuclear membrane, nucleolus, and the chromosomes are not condensed and the centrioles are duplicated. Now as the early phase is followed by the late phase, in late prophase, the nuclear membrane disappears. It integrates and it disappears gradually. And the chromosomes are condensed. Now as these chromosomes are condensed, you can see the chromatids which are attached to the centromeres. Now as we have drawn the structure of the chromosome in this way. Now the point at which the chromosomes are attached to each other, that is called as centromere. This point is called as centromere. So these chromatids are visible under a light microscope in the late prophase. They can be seen, and these sister chromatids, they are attached to the centromere. They are attached to each other by the centromere. This is visible in the late prophase. Also the centrioles which are there, these centrioles, they start moving on the opposite poles. And now the prophase is followed by the metaphase. Now as you can see here, the chromosomes, they are completely condensed, and as they are condensed, the sister chromatids and the centromere, they become very prominent. It means they are visible. So the condensed chromosomes, they fully become condensed, or the condensation of the chromosome is completed over here, and therefore the chromosomes, they appear short. But even if they appear short, they become very prominent. That is, the sister chromatids and the centromere can be visible. They become prominent. Now what happens? The centrioles, which I said, they move towards the opposite poles in the cell. So these two centrioles, from these two centrioles, these strand-like structures you see here, these strand-like structures, they arise, and these are called as the spindle fibers. So from these centrioles, the spindle fibers arise, and these spindle fibers, they get attached to the centrioles of each chromosome, each condensed chromosome. these spindle fibers, they get attached to the centrioles of the condensed chromosome. Now what happens further is these spindle fibers, they start to pull the chromosomes. They start to pull and push the condensed chromosome, and because of this pushing and pulling action of these spindle fibers, the chromosomes are brought in the middle of the cell or in the center of the cell in an equatorial plane. This is called as the metaphase plate. When these chromosomes, they come in the center of the cell, that is called in the equatorial plane. This is called as the metaphase plate. So the pushing and pulling action of the spindle fibers, they bring the chromosomes in the center of the cell. This center of these chromosomes, they appear in an equatorial plane, and that's why it is called as the metaphase plate. It happens in metaphase, that's why it is called as metaphase plate. Now further, what happens due to this pushing and pulling action of the spindle fibers, these centromeres which are there, to which the sister chromatids are attached to each other, these centromeres, they break down into two. They get divided into two, and each centromere has a chromatid with it, and they pull these chromatids towards the centrioles or towards the poles of the cell, towards the opposite poles of the cell. And as the chromosomes are pulled, the centriole gets divided into two, and as the centriole gets divided into two, the chromatids are separated. Suppose this is the spindle fiber which is attached to the chromosome. This is the chromosome and the centriole which is attached to it. Now as the pulling action takes place, from both the poles, these sister chromatids are separated and the centriole gets divided into two. Along with this centriole, the spindle fibers, they pull the sister chromatids at each pole, each separate pole. And now the metaphase is followed by the anaphase. In anaphase, what happens? The spindle fibers which come out of the centrioles from the opposite poles, those spindle fibers, they start pulling the chromosomes, and as they pull the chromosomes towards the opposite poles, the centromere to which the chromosomes are attached or to which the sister chromatids are attached, those centromeres, they get divided into two and they get pulled towards the opposite poles. As these centromeres, they get pulled towards the opposite poles, the chromosomes or the chromatids, they get separated from each other, and each centromere has two chromosomes. So these chromosomes, they get pulled towards the opposite poles, and as these chromosomes or chromatids, they appear to be pulled towards the opposite poles, here this anaphase ends and the daughter chromatids or daughter chromosomes are produced over here in the anaphase. And now the anaphase is followed by the telophase. Telophase is the last stage or the last phase of the karyokinesis process. Now here in the telophase, what happens? The chromosomes which are coiled together or condensed together, which appear to be short. Here what happens in the telophase, those chromosomes, they start uncoiling. They start uncoiling themselves, and again they become long, or again they lengthen their size or their length, and they become long chromosomes. And slowly the nucleus starts to appear, and as the nucleus appears, the nuclear membrane also starts to appear. The nuclear membrane appears, the nucleus reappears, and these centrioles, they move to the opposite poles, and the spindle fibers which came out of these centrioles, these spindle fibers, they get absorbed in the cytoplasm. The spindle fibers, they get absorbed in the cytoplasm and they disappear. And the cytoplasm then tends to divide. Constriction is seen in the cytoplasm. It tends to divide and the two separate nucleus appear in the cell. After the telophase, the second process of the cell division starts, that is cytokinesis. Now, as we know, cyto means which refers to the cytoplasm of the cell, and kinesis means division. So, here in this process, the division of cytoplasm takes place. Now, as the constriction which appeared in the telophase, that constriction, it gets deeper and deeper, and finally in the cytokinesis process, the cell is divided into two daughter cells, two individual daughter cells which are similar to each other. They have equal number of genetic material, they have equal number of chromosomes, equal genetic material distributed among both the cells. Now, in the cytokinesis process, when this process happens in the plants, plant cell wall is very flexible, so it does not support the constriction, or it does not show the constriction. Instead of the constriction and all this, the cell wall, it appears or it gets divided from the middle. For example, suppose this is the plant cell. Now, if the division in the plant cell has to appear, the constriction which appeared in the animal cell, it does not happen like this. A plate is formed in the middle of the cell. This plate is called as cell plate. This plate is called as cell plate, and it appears and it eventually divides the cell into two new daughter cells. Now, this constriction and this division of the cell wall, of the cell and the cytoplasm, or the nucleus and the cytoplasm, and distribution of the genetic material and chromosomes evenly in both the daughter cells is the speciality of animal. During the process of cytokinesis, or at the time of cytokinesis, what happens? The cell organelles like mitochondria and ribosomes and other cell organelles, they are evenly distributed in both the daughter cells. As before, we discussed about the general characteristics of the mitosis process. That is, the mitosis process is a process in which a cell divides to form two similar daughter cells, and it equals, or it distributes the genetic material equally in both the daughter cells. And this type of division is seen or is found in the somatic cell. Somatic cells are the cells of all our body except the reproductive cells. And this mitosis process is also called as equational process, because the chromosome number is evenly distributed in both the daughter cells, or the chromosome number is equal in the daughter cells and their parent cell, equal to their parent cell. So, this is the process of mitosis.